Hello, everyone, and welcome to today's live webinar, The Role of Immune Cells and Immune Checkpoints in Multiple Myeloma, presented by Nathalie Heider-Hönatsch, PhD candidate, University Hospital, Bonn, Germany. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com/cell culture heroes. Now let's get started. Today's webinar is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on that support tab found at the top right of your presentation window, or use that Ask a Question box to let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab and follow the process to obtain your credits. I now present today's speaker, Nathalie Heider-Hönatsch. Nathalie is currently a PhD student at the Department for Oncology, Immunology, and Hematology at the University Hospital Bonn in Germany. She is working in the Biomedical Research Laboratory under the direction of Professor Dr. Peter Brossart. Her thesis focuses on immunotherapy in multiple myeloma. Currently, she investigates the regulation of immune cells and the expression of immune checkpoints, for example, by using CRISPR-Cas9 technology and flow cytometry. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Welcome, Nathalie. You may now begin your presentation.
Yeah, thank you very much for the kind introduction. So my name is Nathalie. And first I want to thank GIBCO for giving me this great opportunity to discuss our work. And of course, I want to thank the University Hospital Bonn and my PI for the support. Okay, so with that, I want to start the talk. I would just mention that it is a really broad overview of the topic. So if anybody has specific questions, please get in touch with us, and I would love to discuss the research in more detail then. Well, let's start with the timeline of historic events in cancer treatment. Everything began with surgery, whereby these treatments were not that accurate and successful alone. After the development of radiotherapy in the early 1900s, the treatment strategies were essentially improved by the discovery of chemotherapeutic drugs around 1940. So these are cytotoxic drugs that mainly aim to kill highly proliferative cells, such as cancer cells. A breakthrough in the field of medical oncology was the development of targeted therapy, like inhibitors or monoclonal antibodies against specific targets on the tumor. So this approach is much more directed and therefore enables more effective tumor cell killing and less side effects. The last turning point in modern oncology took place in 2010 with the use of immunotherapeutic approaches for advanced and metastatic tumors. So this huge and promising fourth column of cancer treatment was established by the work of Ellison and Honjo, I think in the 90s, and instead of targeting the tumor cell itself, immunotherapy aims to boost the immune system and restore its ability to recognize and destroy the cancer cell. In one of the latest GIBCO webinars, we heard also interesting lectures of immunotherapy, for example, the approach of using oncolytic viruses by Alex Baker. So today we want to focus a bit more on the tumor immune microenvironment and immune checkpoint inhibitors. How does it come to tumor progression? A homeostatic immune microenvironment is important to control any alterations arising in the different tissue compartments of the body. So this condition is termed as immunosurveillance, when all the different immune cell populations constantly control the microenvironment and respond targeted to any new upcoming event. Cancer immunoediting describes the process of a shift from such a controlled homeostatic immune microenvironment to immunosuppressive tumor-tolerant microenvironment. This is because of a progress in which immune cells, stroma cells, and tumor cells are in constant interaction, either by direct cell-cell contact or because of their different secreted cytokine and chemokine profile. For the process of tumor cell elimination, it is of high importance that the immune cells see and recognize the malignant cell, especially neoantigens, which are cell structures from these transformed malignant host cells, or are important for this recognition. But when time goes by, tumor cells adapt. They downregulate these antigens and receptors that are recognized by the immune cell, and on the other hand, they also communicate them that they are tolerable and not dangerous. So this state of tumor dormancy contributes to the selection of tumor cell variants with highest capacities to survive, like tumor stem cells. And finally, the suppression of the immune system and the survival of the cancer clones which are fittest contributes to the tumor's escape mechanism. So how does the tumor immune cell interaction look like in more detail? For a full activation of a T cell, which are the main effector cells of the adaptive immune system, 3 signals are actually necessary. So first, the T cell receptor recognizes a specific antigen, for example, an altered structure from a malignant host cell, so that the T cell receptor engages with a major histocompatibility complex receptor from either another immune cell or an antigen-presenting cell, or also from a stroma cell itself. As a control mechanism, various either co-stimulatory or co-inhibitory receptors are then transiently upregulated. So the overall input of these positive or negative signals then determines the activation state of the T cell. They can, for example, develop into an effector T cell or, in contrast, into an immunosuppressive regulatory T cell. And finally, also the cytokine secretion as a feedback loop serves as a fine-tuner for the T cell response, like is it strong or weak, communication to surrounding cells, and so on. And although these co-inhibitory signals are quite important to mediate tolerance and also prevent autoimmunity, tumor cells often use these checkpoints to downregulate the immune response. So one mechanism that tumor cells use to hide and escape from the immune system is to upregulate immune checkpoints like PD-1 and PD-L1, and therefore facilitate a pro-tumorigenic microenvironment. So immunotherapy aims to boost the immune system and restore the ability to recognize cancer cells and kill them. Hereby, a key to success of immunotherapy is the ability of the immune system to actually see the cancer cell. In solid tumors, there is a large discrepancy of immunotherapeutic success described by the model of hot versus cold tumors. So, for example, melanoma is a so-called immunologically hot tumor. They are characterized by a high mutational load and high production of neoantigens, and therefore a massive infiltration of lymphocytes. So they work good for immunotherapeutic approaches, which boost the anti-tumor immune response, and, for example, in combination with radio or chemotherapy, which aim to destroy the target cell and consequently produce even more neoantigens, that makes the tumor more recognizable for the immune cell and increases the cancer cell killing efficiency. But in contrast, so-called immunological cold tumors, like pancreatic cancer, are protected by surrounding stroma cells. So they, for some reason, don't provoke a strong immune response, and T cells are unable to penetrate the tumor. Furthermore, these tumors attract immunosuppressive cells like Tregs or myeloid-derived suppressor cells that furthermore secrete cytokines to downregulate the immune response. And as a result, we get a so-called immune desert which is built around the tumor. Okay, so let's come to the hematological malignancies. My PhD project is about immunotherapeutic approaches in multiple myeloma. Therefore, I would like to give you a brief overview of this malignancy. Multiple myeloma is the second most common hematologic malignancy and occurs at an average age of about 65 years, and it is still an incurable disease. Myeloma is a disorder of terminally differentiated plasma cells characterized by clonal expansion of a malignant plasma cell in the bone marrow and a massive production of immunoglobulins. So these antibodies, which are also called paraproteins, have lost their function and they don't recognize any target structures of, for example, invading pathogens or aberrant host cells anymore. Instead, they accumulate, and so this uncontrolled proliferation of unfunctional plasma cells replaces the normal, healthy hematopoietic cells in the bone marrow. The so-called CREB criteria are essential for the diagnostic of myeloma. So first, elevated calcium levels are caused by the altered bone metabolism, with increased bone resorption. And then there is an impaired kidney function, which arises because of the massive production of monoclonal immunoglobulins in the blood. The kidneys try to filter these antibodies, but the accumulation of these paraproteins increase with the disease progression. Also, anemia and infections are a problem because of the infiltration of the tumor cells in the bone marrow that suppress the normal hematopoiesis. And finally, main difficulties are made by the bone lesions. So the bone material is not static. It is actually a constant progress of bone resorption whereby the osteoclasts resorb the material and that the osteoblasts gain the bone material. And the bone pain of myeloma patients is mainly increased due to the increased osteoclastogenesis during this progress. So the initial genetic event is thought to occur in the germinal center of the lymph nodes where B cells mature. Facilitated by somatic hypermutation and isotype switching, a founder clone arises, and due to the novel mutations, time by time, this disease progresses to a first still asymptomatic state called monoclonal gammopathy of undetermined significance, which is MGUS, and then proceeds to smoldering myeloma and finally to the symptomatic myeloma. So a main feature of this disease is the high heterogeneity represented in these different subclones, making the patient treatment more challengeable. At the bottom of the slide, you can see general biological events typical for the disease progression. Like in other cancer types, the tumor cells become more aggressive and the tumor microenvironment shifts to a tumor-friendly surrounding. But typical for myeloma, as mentioned, is the altered bone metabolism. Okay, don't be scared because of this full slide. It's actually one of my favorites. So in the corner, you can see some of the typical hallmarks of cancer. The altered bone metabolism with increased osteoclastogenesis, leading to the typical bone lesions is mentioned at the upper left. Key signaling axis here is the rank-rankle signaling pathway. In contrast, increased vascularization is a quite common hallmark of cancer. The accelerated proliferation of cancer cells is linked to an increased metabolism, and the need of more nutrition and oxygen is also a common hallmark. And this adaptation is facilitated by neoangiogenesis, the formation of new but also weakened blood vessels. The alteration of the tumor stroma microenvironment, you can see it in the corner on the left-hand side, is key for the development of cancer. Similar to the immune cells, the stroma cells are in constant contact with the tumor cells, communicate and influence each other. So also the extracellular matrix of the cells changes, and they begin to secrete pro-tumorigenic factors like survival factors, growth factors, and other stimulative cytokines. So we want to focus on the shift from an anti-tumor to a tumor-tolerant microenvironment. As mentioned previously, the upregulation of immune checkpoints contributes to the tumor immune escape mechanism. Sorry. In the case of myeloma, the PD-1, PD-L1 means programmed DEF1 and programmed DEF ligand-1 signaling axis is especially critical, and there are different immune checkpoint inhibitors in clinical trials, but they are not sufficient as monotherapy. Okay, and in the center you can see this pinkish malignant plasma cell, and surrounded different treatment modalities ongoing to target the myeloma bone marrow niche. And the first-line therapy strategies today is the combination, or I hope I pronounce it right, if not, sorry, dexamethasone with different immune modulatory drugs like lenalidomide and the proteasome inhibitor bortezomib. Well, for the case of advanced stage diseases like relapse and refractory cases, there are clinical trials for the combination of these drugs and boosting them with different immunotherapeutic strategies like monoclonal antibodies mediating, for example, antibody-dependent cytotoxicity, or different vaccination strategies using dendritic cells. So, as patients with advanced disease stage show high upregulation of PD-1 and PD-L1 in bone marrow and blood tissue samples, we want to focus on this immune checkpoint. So myeloma is still incurable, and monotherapeutic approaches are not sufficient. Therefore, we aim to find a beneficial immunotherapeutic approach based on immune checkpoint inhibition. These immunotherapeutic approaches can be divided into three different strategies. Agents that boost the immune response by blocking and reversing immunosuppressive pathways, like immune checkpoint inhibition, or drugs that specifically target the malignant clone, for example, receptors that are only expressed on the tumor cells and then mediate cytotoxicity, like monoclonal antibodies targeting BCMA or SLAM-F7 on the malignant clone. Or very personalized and promising cellular-based therapy approaches where mostly autologous immune cells are modified to achieve a much higher anti-tumor response like CAR-T or CAR-NK cell therapy. So as mentioned, the PD-1 and PD-L1 immune checkpoint is upregulated in myeloma patient samples of advanced disease stage, which make this pathway to a promising target. During the immune cell-tumor cell interaction, it is not only the problem that these co-inhibitory receptors transiently downregulate the immune response. In a state of chronic inflammation, similar to a chronic viral infection, the immune cell became exhausted. That means they are constantly low activated, but are not responding in a proper way anymore, and instead become apoptotic after a while. So you can see in this illustration that if a T cell comes in contact to a tumor cell via the antigen presentation, the upregulation of PD-1 abrogates the downstream T cell receptor signaling pathway. And instead of producing pro-inflammatory cytokines like IL-1, TNF, or interferon, they start to secrete immunosuppressive cytokines like interleukin-10. Therefore, they not only differentiate into an anergic, exhausted, and immunosuppressive phenotype, they also influence the tumor immune microenvironment with these pro-tumor cytokine milieu. So in my PhD project, we aim to knock off the signaling pathway and to investigate the influence of other immune and stroma cells. To do so, we work with the mouse myeloma cell line 5T33, which not only enables in vitro studies, also there's an established mouse model with the use of the cell line. So this 5T33 mouse myeloma cell line strongly expresses PD-L1, and we use the CRISPR-Cas9 technology to deplete this gene. There are many different methods to deliver the CRISPR plasmid into the target cell line, but as these 5T33 cells are suspension cells, electroporation seems to be the most efficient way for the transduction. At the bottom, you can see the organization of a typical CRISPR-Cas locus in a microbial genome. So CRISPR loci contain a combination of CRISPR-associated genes called Cas, then the endonucleases, and as well non-coding RNA elements, which enable the specific cleavage of furin nucleic acid. The mechanism of the CRISPR-Cas system can be divided into 3 stages. So first, in the acquisition stage, the conserved Cas endonuclease complex acquires a specific sequence, the protospacer, from the foreign invader DNA and integrates it as a new spacer into the CRISPR array, which then later serves as the immunological memory. And second, in the expression stage, the CRISPR array is transcribed into a precursor RNA transcript that is further processed into small units of RNA known as CRISPR RNAs. So these CRISPR RNAs are combined with the Cas protein to form the active Cas-CRISPR RNA ribonucleoprotein complex, whereby the CRISPR RNA functions now as a guide for the endonuclease to identify complementary structures. And third, in the interference stage, the CRISPR RNA Cas ribonucleoprotein complex, very nice word, then scans the cell for new foreign nucleic acid targets, which are recognized by complementary base pairing. And a successful recognition then leads to the cleavage and degradation of the target nucleic acid. So the guide RNA determines where the insertions or deletions in the gene will occur. And once the guide RNA and Cas9 protein are expressed in the cell, the guide RNA will direct the Cas9 to bind the target sequence and introduce a double-strand break. The cell can then repair the break either with non-homologous end joining or with homology-directed repair mechanism. Non-homologous end joining is the most active repair mechanism, but often leads to insertions or deletions near the target sequence. If these so-called indents occur within an open reading tray, it may introduce a frame shift that causes a premature stop codon, eliminating the gene function. Therefore, it might be beneficial to add a homology-directed repair template to enable specific gene repair. And furthermore, these homology-directed repair plasmids can contain additionally, for example, a reporter gene, a red fluorescence reporter gene, or resistant against antibiotics like pyrimycin, so enabling better cell selection. But to be honest, especially for suspension cells, it is very hard to achieve such a co-transfection. So we continued our work with the 5T33 PD-L1 knockout cells, which are already transduced with GFP. Okay, now what to do with this immune checkpoint blockade? In our lab, we use that cell line to characterize the myeloma cell independence of the PD-L1 signaling axis, the behavior under different drug treatment, and of course, we also want to investigate the tumor immune cell interactions. But the most exciting part is to be continued, as that fascinating research is still in progress, and we are looking forward to present you our interesting findings in the next publication. Till that, I would like to highlight the work of this working group, which analyzed the tumor immune microenvironment during myeloma disease progression using the 5T33 myeloma mouse model. They nicely show that the amount of PD-1 positive CD4 and CD8 T cells increases with the amount of PDL positive plasma cells in the bone marrow and in the spleen of tumor-bearing mice. And furthermore, these PD-1 positive T cells additionally co-express other immune checkpoints, in this case TIM3, and were also partly apoptotic compared to the healthy control. So this working group did also functional assays with these exhausted immune cells and identified the loss of effector cell functions and an increased immunosuppressive phenotype measured by the altered cytokine secretion. So these findings as the base gives insights into a very complex, highly regulated tumor immune microenvironment and gives hints that there are many more other alterations during the disease progression to be identified. With this open end, I want to close my talk, and I wish any researchers also working hard on the optimization of cancer treatments all the best of luck. Okay, so in summary, we learned how the tumor cells will influence their surrounding stroma and immune microenvironment, a mechanism called cancer immunotherapy leading to tumor escape. And we clarified the function of immune checkpoints like PD-1 as signaling axis on which T-cell responses are modulated, followed by the development of T-cell exhaustion. And furthermore, we focused on multiple myeloma, which is still an incurable disease with high need of new treatment modalities. PD-1 and PD-L1 is highly upregulated in patients of advanced disease stage. Immune checkpoint inhibitors are in clinical trials, but they are not sufficient as monotherapy. And instead, we need a combination with other strategies to boost the immune system and to make the tumor cells more visible for the immune system. And we talked about the microbial defense system CRISPR-Cas, which is mediated by 3 different steps: acquisition, expression, and interference. And this is a system which makes us possible to delete and insert any gene of interest. And finally, we talked about the identification of cell surface markers and cell populations using flow cytometry and a mouse myeloma model using 5T33 cells. Okay, and this is the literature I used, but there are many other great populations for this exciting field of research. Okay, and with this, I want to thank my working group, my supervisor. It is such a great time, and I'm very thankful for the opportunity to work in this lab with these great people, and actually I almost learn every day. So thanks to Uni Bonn. And of course, thank you, GIBCO, for giving me this nice opportunity to talk about our research field. Yeah, and last but not least, thank you so much for listening, especially because it was a really broad talk. I'm very happy to discuss any more details with you, so please feel free to ask questions and keep in touch. Thank you very much.
Thank you, Nathalie, for your informative presentation. We will now start the live Q&A portion of our webinar. Now to our audience, if you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. So we have some great questions already coming in. Nathalie, let's start with this one. How do we achieve a better cancer treatment in the future?
Yeah, this is, of course, a very good question. And I'm sure I have not all the answers, but one part is definitely the development to personalized medicine. So as surgery, chemotherapy, and radiotherapy are working like the model of one drug fits all, the use of cancer immunotherapy is much more personalized to the individual tumor. So while cytotoxic drugs aim to kill just highly proliferative cells, immunotherapeutic approaches boost the patient's own immune system with a variety of targets. So precision medicine aims to diagnose actually the disease before it clinically outbreaks so that the treatment can start as early as possible to control the disease. But for this approach, of course, the diagnostic methods has to be very improved, like all the omics studies. But I think the development is going more and more into this direction. But it's quite complicated, of course, to generate all these data, like finding biomarkers for the diseases. And we not only have to do a lot of clinical studies to find these markers, but also use these big data in the right way. And in general, I think the strategies for hematological malignancies seems to be that there's a tendency of, more like 3 treatment strategies are better than 2, or in the future, I don't know, maybe 4 are better than 3. So monotherapeutic approaches are mostly because of the high redundancy, not that effective. Yeah, this is, I think, in general, the tendency. And for example, as a specific point, as mentioned, turning cold tumors like prostate cancer into immunological hot tumors will also be a challenge because of the trafficking and homing of the immune cells that actually cannot reach these tumors. So if that could be improved, I think it's going to be much better then.
Thank you, Nathalie. And here's another great question. What is CAR T cell therapy? Is it already in use?
So CAR T cell therapy is a type of adaptive cell therapy using engineered T cells to specifically target a receptor highly expressed on a tumor cell. So, these cells have a chimeric antigen receptor, which consists of the T cell receptor, of course, that recognizes a target, and it is also included an intracellular activation domain, or I think CD3 and CD28, so that the full activation of this T cell is all the time active and enables the target cell killing without any MHC molecule. So I think it's also already in use, for example, for myeloma, ALL, CLL, but as far as I know, only for refractory or relapsed patients. So for example, in B-cell lymphomas, CD38 and CD19 are often targets, and this CAR T cells then directly kill especially these cells, but of course also healthy tissue expresses these markers, so there's some kind of risk, or also developing resistance. But this is already showing promising effects. So basically, the T cells have the specificity of antibodies, the effector function of cytotoxic T cells, but are not dependent on other stimuli and then target and are not prone for immunosuppressive mechanisms by the tumor microenvironment.
Now, Nathalie, are there other immune cells that are regulated by immune checkpoints besides T lymphocytes?
Absolutely. So the function of immune checkpoints on T cells is much best known on T like CD4 and CD8 T cells, but for example also NK T cells are tightly regulated by co-inhibitory receptors. But although their function is actually completely different. So, for example, NK cells scan surrounding cells for their MHC expression pattern, and if they cannot detect the MHC complex, they unleash their killer cell function. So this mechanism enables then the differentiation between self and non-self, or for example on affected T cells by viruses, for example, and the responsible receptor for this is the key receptor, which is also a co-inhibitory receptor. So, but also other checkpoints like PD-1 and CTLA-4, TIGIT, and VISTA regulate NK cell function, and of course there are also checkpoint inhibitors under investigation. Also, interestingly, immunosuppressive cells like Tregs and myeloid-derived suppressor cells also express inhibitory receptors. So, yes, it's a general overall phenomenon.
Now, Nathalie, have you identified the genes that you would introduce or delete to increase the chance of remission?
No, not so far. This is really a good question, but it's not very easy to identify. Of course, always we look for these genes which are highly upregulated during the disease, and we focus on the PD-1, PD-L1 pathway, but for this, for example, next generation sequencing could help, and FACS staining, so flow cytometry analysis for these receptors which are upregulated. But we are looking on this, and we have to see. But it's an interesting point, and I cannot explain it yet because the publication is not here.
Thank you, Nathalie. Now our next question, does hypoxia play a role in myeloma or the treatment?
Oh yeah, absolutely. So while in solid tumors, hypoxia is built due to the increased proliferation and tumor growth, where neoangiogenesis mostly promotes insufficient weakened blood vessel formation, the oxygen level shrinks below normoxia, and this is only for the solid tumors. But in case of the myeloma, the bone marrow itself is a hypoxic niche. So the myeloma cells constantly activate, for example, HIF-1 alpha, and therefore the hypoxia responsible genes can promote survival, resistance, and proliferation, and the tumor cells can grow. But in contrast, the T cells, like effector T cells, cannot live within this hypoxic level. Actually, interestingly, not because only of the less oxygen, more because of the acidic pH. And they cannot adapt, so the T cells go into apoptosis, and instead immunosuppressive cells like Tregs are stimulated. So this is really a point in myeloma. And the trick is that there are hypoxic sensitive drugs which are not working under nymoxia. They stay as a prodrug, and in hypoxia they convert into their cytotoxic form and therefore mainly destroy the tumor cell there.
Thank you, Nathalie. Now here's an interesting question. In your opinion, is there any scenario where immunotherapy could cause a kind of autoimmune condition or disease?
Absolutely. This is a point. So whether it is immune checkpoint inhibition or CAR T-cell therapy, there can always be some adverse effects. Yeah? So if we block the immune checkpoints, which are responsible for self-tolerance, of course, there's the risk that the immune cell, the immune system targets itself. So it's already been described that some patients which got, I think it was immune checkpoint inhibition, also develop rheumatological diseases like arthritis, and this is always a risk. Also, the cytokine release syndrome, which is a systemic, strong inflammatory response, can cause similar diseases. So, yeah, that's happened indeed. But the treatments are going better and better, and it's more possible to predict such diseases. So it's still a risk, but it's developing in a good direction, I think.
Thank you, Nathalie. Now, our next question is two parts. So I'm going to read the whole question, and we can answer it part by part. And we have a question coming in from Sebastian. Dear Nathalie, thank you for this comprehensive overview. I actually have two questions. Maybe I did not get it during your talk. Number one, how about metastasis in hematologic entities? Is it possible, or can it be described in a different way? And also, are there forms of metastasis known in multiple myeloma? You mentioned osteoblasts and osteoclasts. Thank you very much. I'll let you start with the first part.
Yeah, so the point of metastasis in hematological diseases. So we know that in solid tumors, of course, the cancer cells need to transform. So during the progress of EMT met formation, which is epithelial mesenchymal transition, the stroma cells just first have to gain capabilities to migrate and leave their primary tissue. So the first steps of migration, invasion, and internalization into the blood or lymphatic system, this is obviously not necessary for hematological tumors, as they are already circulating in the blood. But yes, the other steps, as far as I know, are quite comparable. So in the vascularizing system, the tumor cells have to escape from the immune attack, which is one point, and at the distant side, the need to extravasate into the new tissue. And in case of hematological cancer cells, these are often secondary lymphoid organs like spleen, lymph nodes, maybe the liver. And it's their natural function actually for generating immune responses. They don't need a lot of mutations to acquire such a function, but it is still, I think, a critical step, as in solid tumors, to adapt at secondary sites to build new colonies in a different microenvironment. So I can imagine it is kind of a metastasis, yes, quite comparable. And what was the second question? If the tumor cells destroy the bones itself. So, well, actually, it's more an indirect effect. So a hallmark of myeloma is the increased osteoclast activity. In normal cases, osteoclasts resorb the bone material, and osteoblasts promote the growth. So this is they are regulating itself via the rank-rankle signaling pathway. And in the myeloma, this is really disrupt this progress, and the myeloma cells activate this rankle signaling and promote the osteoclastogenesis. And as a consequence, the bone material is destroyed, and this leads to the secretion of growth factors and also interleukin-6, which in turn stimulates the myeloma cell proliferation. So this is like a bad feedback loop. And interestingly also, I think monocytes and macrophages can be also stimulated by this rankle OPG signaling pathway and further differentiate into osteoclasts, also worsening this cycle of osteolytic bone lesions. So it's really a indirect effect of the myeloma cells.
Thank you, Nathalie. Now we have a question coming in from Uma, and she would like to know if you could tell us more about T-cell exhaustion in this instance.
Pardon, I didn't get it.
Oh, sure. Can you tell us more about T-cell exhaustion in this instance?
I cannot understand what is instance. The last word, sorry.
In this relevance, in this instance, in this example.
Okay. So you mean in the PD-1, PD-ligand-1 pathway in myeloma, or what is actually the focus? So exhaustion of T cells is a step further than just being downregulated. So it is normal that the receptor, the co-inhibitory receptors, are upregulated and then downregulate the immune response, right? And just as they are constantly downregulated, like in viral infections or in the tumor progression, these continuously upregulation of co-inhibitory receptors leading to the exhaustion. And this is an analgesic state where the T cells don't even respond to anything anymore, and instead they go into apoptosis. So they are not only downregulated, they die more or less after this stage. So, this is actually killing.
Thank you, Nathalie. We have so many great questions coming in, but we only have time for one more question. And I do want to remind our audience that those questions we do not have time to answer today, and additionally those questions that are submitted during the on-demand period, they will be addressed by the speaker via the email address that you provided at the time of your registration. Okay, Natalie, wrapping with this question, how specific is immunotherapy? Is it really safe? And what are the risks?
Yeah, well, we already came to this point. So it is more or less specific, as we always try to find a target which is mainly expressed on the tumor cell, right? But always, like in B-cell leukemias, they, for example, myeloma, where the target cell is the plasma cell, these markers like BCMA or CD38 are also expressed on the healthy cells and sometimes on the surrounding tissues. So, as already said, there's always the risk of on target but off tumor effect, so they say. And yeah, this is why it is to be optimized, and we are still searching for more specific targets, but at least there's always a risk to develop like the cytokine release syndrome or any other problematic features that target the healthy tissue of the patient or host.
Thank you, Natalie. And thank you for today's Q&A. This was great, very detailed answers. Do you have any final comments for our audience?
Well, actually just saying thank you, GIBCO, LabRoots, and Thermo Fisher for this nice communication platform. So I think it's very important to communicate the science, not only to connect the scientists, also to connect with the world. And I think it's a very good idea. And yeah, thanks a lot. Also for these nice questions. They are very, not that easy, but very intelligent. Thanks.
Thank you again to our Nathalie Heider-Hönatsch for her time today and for very important research. Now, I would like to thank LabRoots, but I also want to thank our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. You will now be redirected to our next Cell Culture Heroes webinar, the role of SIRT1, LXR signaling, and diabetic retinopathy, presented by Sandra Hammer, Research Fellow, Michigan State University. This webinar will broadcast live on December 17th at 9 a.m. Pacific time. We hope to see you there, and bye for now, and have a great day.